All right, so get ready.
Okay.
Because today we're diving deep into the world of Eastern equine encephalitis.
Oh.
Exactly. Yeah. We're going to take a real technical look at this virus from especially like a neurologist's perspective.
Okay.
This was one of the first neuroinvasive arboviral diseases recognized in North America.
Right.
But there's a lot of evidence that suggests that its prevalence and public health burden are changing.
Interesting.
So today is really going to be for those of you out there that want like a deep, really technical understanding of EEC.
That's great. And it's a really important topic, especially given, the changing patterns we're seeing. So understanding this can really help the medical community, but also, people who just like to be outside.
Exactly.
That's all of us.
That is all of us.
Yeah.
And we have a whole stack of research articles and clinical data that we are going to break down for all of you so that we can make the complex clear.
Sounds good.
So to start off, where does EEV, the virus itself, fit into the world of viruses?
So it is classified as a group 4. In the Baltimore classification system, meaning it's a positive sense single-stranded RNA virus.
Okay.
And even more specifically, it belongs to the alpha virus genus within the Togaviridae family.
Wow.
And so that's how we classify it, really based on the genetic makeup and how it replicates.
So it's a pretty sneaky virus, right?
Yeah, it's a sneaky one.
It's efficient.
Very much so.
But, I remember reading something about an older classification system.
Oh, yeah, you're right. Yeah.
Yeah. Can you just remind me, what was that?
So there was the older system that grouped arboviruses into category A, B, C, and D. And this was really based on the genus.
Okay.
So alpha viruses were group A, flaviruses were group B, so on and so forth.
Okay.
But the Baltimore system is really the gold standard.
Okay.
Because it gives us a clearer picture of how they operate, and that is based on, as we were saying, the genome and the replication strategy.
Got it.
Yeah.
Okay, so let's zoom in then on the EEV genome.
All right.
What can you tell us about its structure? What's encoded in that genome?
Yeah, so the EEV genome is about 12 kilobases long.
Okay.
And it encodes 9 proteins.
Right.
5 of these are structural, so meaning they form the actual structure of the virus. You can think of it like the building blocks.
Yeah.
And then 4 of them are non-structural.
Okay.
Which is essentially the machinery that the virus uses to actually make copies of itself.
So it's not just about invading.
Yeah.
It's about replicating.
Exactly. It's about replicating. It's about spreading.
Wow.
And the really cool thing is that these proteins are encoded from different regions of the genome.
Okay.
The structural proteins are encoded from the 3' end through a subgenomic RNA transcript called 26S.
Okay.
And the non-structural proteins are encoded from the 5' end.
Got it.
From a full-length genomic RNA.
Okay, that's fascinating. How much can be encoded into such a small space?
Absolutely, yeah. And all this is happening on a microscopic level. It's pretty fascinating.
Okay, so can we clarify then what those structural and non-structural proteins actually do?
Of course. So the structural proteins really form what we consider the outer shell and the weapons.
Okay.
So they form the capsid, which is that protective coat around the genetic material.
Okay.
And the envelope proteins, which are critical for latching onto and invading the host cell.
So it's like this little armored vehicle that's ready to,
deliver the payload.
Yeah, deliver the payload.
Yeah, exactly.
Okay. And then the non-structural proteins.
So the non-structural proteins are the ones that are responsible for replicating the viral genome, producing new viral proteins, and then assembling those new viral particles.
So they're kind of running the show inside the cell.
Yeah, they're the ones that are really taking over the machinery.
That's incredible.
Yeah, it's really remarkable.
That we know all this stuff now about these microscopic processes.
I agree.
You also mentioned the formation of E2, E1 heterodimers.
Yes.
What are those?
So initially, these structural proteins are produced as one long chain.
Okay.
And this long chain actually gets processed and cleaved within the host cell to form these E2, E1 heterodimers, which are essentially 2 different proteins, E2 and E1, bound together.
Okay.
And these are the key components of what we call the spike proteins
that
protrude from the actual viral particle.
So the spikes are how it actually grabs onto and enters the host cell.
Exactly. They're the key.
They're like the like the grappling hooks, right?
Yeah, exactly. And the E2 protein specifically is the one that's really critical for binding to the host cell receptors. And it does this through its lysine residues at positions 71, 74, and 77.
Oh, wow. So specific.
Very specific.
So, okay, we got this, general structure down.
Yeah.
But aren't there different subtypes of EEV?
That's right.
Yeah.
So in the Western Hemisphere, there are 4 subtypes. Subtype 1, known as NAEEE.
Okay.
That's what we find in North America and the Caribbean.
Okay.
And then subtypes 2, 3, and 4.
Okay.
We call that SAEEV.
Okay.
Which we find in Central and South America.
So there's diversity even within
there is.
EEV?
Absolutely.
Do these subtypes, do they behave differently?
They do. The North American subtype is actually highly pathogenic.
Okay.
Genetically stable.
Okay.
Whereas the South American subtypes are less pathogenic and show more genetic diversity.
So when we talk about like E being this big health threat, it's really the North American subtype.
Exactly. And actually, the South American subtypes have now been reclassified as a separate virus called the Madariaga virus.
Oh, wow.
Yeah, named after the region in Argentina where it was discovered.
So Madariaga virus is kind of like a distant cousin?
Yeah, a distant cousin that's less likely to cause severe disease.
Got it.
Yeah.
So, but that still leaves us with the North American subtype?
Absolutely.
What makes this one so worrisome?
Well, a lot of things, but one of them is its genetic stability.
Oh, eh?
So recent studies have shown that there's a high level of conservation between the strains, even those from different outbreaks.
Wow.
So this suggests that it's maintained its virulence over time.
So it's kind of like a master criminal who's really
perfected their methods.
Yeah, perfected the method.
Sticking to it. Yeah.
Okay, so we've got this stable, highly pathogenic virus circulating out there.
Yes.
How does it actually spread?
So let's talk about those enzootic and epizootic cycles.
Yeah, okay.
So the enzootic cycle, think of that as the quiet life cycle, the behind the scenes.
Okay.
And this involves a specific mosquito species, Culiseta melanura, and certain birds.
Okay.
Primarily passerine birds, so think sparrows, finches.
Okay.
And these birds act as what we call reservoir hosts, meaning they can carry the virus without really showing any signs of illness.
Okay.
And so the virus is going back and forth between these Culiseta melanura mosquitoes and these birds.
So it's like this hidden network where this virus is just quietly circulating among the birds. And then the epizootic cycle is where things get a little more dicey.
A little more complicated,
Yeah, a little more complicated,
So this is where we have the emergence of what we call bridge vector mosquitoes. So these are different species like Aedes, Culex, and Coquillettidia mosquitoes.
Okay.
And they're not as picky.
Okay.
They will feed on a wider range of hosts.
So they're kind of like the crossover artists.
Exactly.
This mosquito world.
Yeah, the crossover artists, exactly. And they can pick up the virus from these infected birds and then transmit it to horses, which can develop severe illness, or us, which we can also...
And that's why E is such a
concern.
No, it's not just, bird disease.
It can spill over.
Yeah, it spills over.
And horses and humans, although we can get very sick... we're actually considered dead-end hosts.
What does that mean?
So that means that if a horse or a human gets infected, we don't have high enough levels of the virus in our blood to actually infect another mosquito.
Oh, interesting.
So we're not really contributing to the spread.
Got it.
The real drivers are the birds and those bridge vectors.
Okay, so the epizootic cycle is kind of like a side road branching off.
Yeah, a side road branching off.
The main highway of the enzootic cycle.
I like that, yeah.
With those bridge vectors being the off-ramps.
Yeah.
And then that risk of transmission is determined by a number of factors, right?
A number of factors, yeah.
Like how much virus is actually in the bird's blood.
The level of virus in the blood, how much the mosquito is biting, the prevalence of infected birds in the area, even the size of the blood meal that the mosquito takes.
That's wild.
It's a lot of different things that all come together.
And have scientists created like mathematical models?
They have.
To kind of predict these epizootic cycles?
Yeah, they've actually developed these models to help us predict these.
Wow.
And it really suggests that these outbreaks are more likely to happen when the infection rates in the reservoir host are high. Okay. When the bridge vectors are actively feeding. Okay. And when there's a lot of overlap between the bird, mosquito, and human population.
So basically, more infected birds, more hungry mosquitoes, and more human-animal interaction equals
greater risk for us.
Yeah, exactly.
But even with all of that, those models suggest that sustained human-to-human transmission is unlikely.
Very unlikely, yeah.
Okay, that is a little bit reassuring.
Yeah.
But it doesn't mean we're off the hook. Right.
No, definitely not off the hook. It's still a serious concern.
Yeah.
And we need to understand its ecological nuances to help us prevent and control it.
So let's talk geography.
All right.
Where in the U.S. typically do we see E activity?
So EEV is primarily found in the eastern United States.
Okay.
And there are some distinct regional patterns.
Okay.
So, for example, in the Northeast, the upper Mid-Atlantic, and the eastern Great Lakes region, we see what's called seasonal intermittent endemicity.
Okay.
So that means that it's there, but it kind of ebbs and flows with the seasons.
Okay, got it.
But in the Southeast, so think Florida, particularly Florida, the enzootic cycles can persist year-round. So if you're living in the Southeast, you might have
a higher
risk
of exposure.
Got it.
Just because it's circulating.
Okay.
Yeah.
So even though EE is, relatively rare, why is it considered this serious public health threat?
Right. So one of the biggest reasons is that it has such a high case fatality rate.
Okay.
So unfortunately, it's one of the deadliest mosquito-borne diseases in the United States.
And so a significant percentage of people that are infected.
The significant percentage will die from it.
Oh, wow.
And even for those that do survive, there's a really high risk of long-term neurological problems.
So even if you do survive this initial infection, you could
have lasting disabilities.
Yeah, that affect your quality of life.
And these disabilities can range from cognitive impairment, personality changes, seizures, and even paralysis.
That's heartbreaking.
Yeah, it's tough.
And the thing about EE is that this transmission occurs
during
when people like to be.
Yeah, the summer, early fall.
Right, when you're outside.
When you're outside enjoying, the weather, the activities.
Exactly.
And, I was also reading a lot about how climate change, biodiversity loss, and human development patterns might actually be influencing this prevalence of EEV.
Right.
Can you kind of elaborate on that a little bit?
Yeah. So warmer temperatures can lead to longer mosquito breeding seasons.
Okay.
And changes in the habitats that the mosquitoes thrive in.
Okay.
And then deforestation and habitat fragmentation can actually concentrate those bird populations.
Okay.
And potentially increase their interactions with mosquitoes.
Got it.
And amplify the viral prevalence.
So it's like a whole bunch of factors all coming together to make this problem worse.
Work together,
So it's not just, isolated factors. It's this whole... Yeah, it's complex.
Yeah, this complex interplay.
It is a complex interplay, and that's why it's so important to understand it.
Yeah.
So we can develop good prevention and control strategies.
Okay, so are there certain populations that are more vulnerable?
There are. The incidence of neuroinvasive disease is higher in men, younger and older age groups, and minority groups.
Okay.
So that's something to keep in mind as well.
So again, it's not just about the virus itself. But it's about all these other
social
and environmental factors.
Social, environmental, exactly.
Okay, so now we've covered kind of the basics about the virus itself, how it spreads, and why it's a threat. But now let's get into what actually happens when EEE invades the body.
Okay.
So can you walk us through step by step that pathogenesis of EE?
Of course.
Yeah.
So it all begins, as you know, with the mosquito bite.
Right.
The virus enters through the mosquito saliva and begins replicating.
Through there at the bite site.
So even before it gets to the brain.
It's making copies.
Yeah.
Making copies, spreading.
And then from there, it spreads through the bloodstream.
Through the bloodstream to other tissues throughout the body.
And for those individuals that do develop encephalitis, the infection progresses in two phases, right? What are those two phases?
So the first phase involves the viral replication in those peripheral tissues and the bloodstream.
Okay.
So it's widespread dissemination.
And that's when you start to feel those like flu-like symptoms?
Exactly.
The fever, the aches.
Yep.
Okay. And then phase two?
So phase two is where things take a turn.
Okay.
It enters the central nervous system, the brain, and the spinal cord.
Wow.
And that's where we actually see high levels of viral RNA and antigen in the neurons.
So it's like this targeted attack.
It is.
On the command center.
Yeah, it's like a hostile takeover of the nervous system. And then that invasion of the central nervous system is what leads to the neurological symptoms of EEEV.
Exactly, those characteristic symptoms.
Okay, but our bodies aren't just,
right.
sitting ducks.
Right, we have defenses.
Yeah.
So our immune system is constantly on patrol looking for anything that's foreign.
Yeah.
And when it encounters a virus like EEV, it launches a multi-pronged attack.
Okay.
And the myeloid cells, which are part of the innate immune system, are the first responders.
So it sounds like EEV might have some tricks.
Oh, it does.
To kind of evade these defenses.
Oh, it's very sneaky. It's a very sneaky virus.
Yeah.
So one of the concerning things about EEV is that it binds to something called heparin sulfate, or HS.
Okay.
Which is a molecule that's found on the surface of various cells, including those myeloid cells.
Okay.
And while a lot of viruses use this to gain entry, EEV actually uses it to suppress the immune response.
So it's not...
It's almost like it's silencing the alarm bells.
Yeah, it's kind of turning off the alarm.
It's silencing the alarm bells.
And it's doing this through, is it the E2 protein again?
It is. It's the E2 protein of EEV.
Wow.
And this is specifically involving the lysine residues at positions 71, 74, and 77.
So this tiny little protein is really playing a big role.
It is.
It's a big role in the virus's ability to evade the immune system.
So we have to understand this molecular level to really target it exactly to block this immune evasion mechanism.
All right. So let's say the virus has managed to evade those initial defenses.
Yeah.
How does it actually get into the brain?
So the most likely culprit is hematogenous spread, which essentially means through the bloodstream.
So it's like hitching a ride.
Yeah, hitching a ride.
In the blood.
In the blood, exactly.
But how does it get through the blood-brain barrier?
So there's a few possible routes of neuroinvasion, and we're still learning about it.
Okay.
But the hematogenous spread is most likely.
Could it, travel along nerves?
So there's a possibility of what we call retrograde axonal transport, which is where it travels backwards along the nerve fibers. And some studies have also suggested that it might actually be able to invade the central nervous system through the olfactory tract.
Wow.
Which is essentially our sense of smell.
That's wild.
But we need to, do more research to confirm this.
Okay, so it has multiple options.
Yes, it seems to.
For getting into the brain.
Exactly, making it that much harder to defend against.
Yeah.
Yep.
Okay, so now it's in the brain. Right. What happens?
Okay, so once it's in the brain, it starts replicating rapidly within those neurons.
Go ahead.
Which are those cells that are transmitting all the signals. Yeah. And this replication triggers inflammation. And cell death.
Wow.
And that leads to widespread damage in the brain.
And is that damage to those neurons? Is that what causes?
That's exactly what's causing those neurological symptoms.
Like the seizures?
The seizures, the paralysis, cognitive impairment, all those things.
Right.
Yeah, and the specific areas of the brain that are affected can vary, leading to a wide range of neurological complications.
Okay, so now we've got the kind of like the cellular level.
Yes.
But how does this present in a patient?
So it's important to note that only a small percentage of people that are infected with EV actually develop severe neurological disease.
Okay.
Many people can experience mild or no symptoms at all.
So it's kind of like the tip of the iceberg.
It is. It's like the tip of the iceberg.
Okay, so, but for those that do develop encephalitis, it's a pretty devastating.
It can be, yes.
Of course, yeah.
So after an incubation period of about 3 to 10 days, the initial symptoms are often nonspecific. They're flu-like.
So fever, headache.
Fever, headache.
Fever, headaches.
Muscle aches.
Muscle aches.
Fatigue, all of those, yep.
So, it can be easy to miss those early signs.
It can be very easy to miss it, especially during mosquito season when a lot of other things are circulating.
Yeah, exactly.
However, in cases of encephalitis, those initial symptoms are usually followed by rapid neurological deterioration.
And how rapid are we talking?
It can be hours or days.
Wow, that's incredibly fast.
And so that's when people may develop altered mental status, confusion, seizures, paralysis, and even coma.
So that rapid progression is kind of
one of the hallmarks
of
E.
Yeah. Are there certain neurological complications?
There are.
That are common?
So while the virus can affect various parts of the brain, some complications are more frequent than others.
Okay.
And these include stupor or coma, seizures, focal weakness, and cranial neuropathies.
Cranial neuropathies, what are those?
So cranial nerves are the nerves that originate directly from the brain. And they control things like our facial muscles, eye movements, and even our sense of smell.
Okay.
And damage to these nerves can lead to things like facial drooping, double vision, difficulty swallowing, and loss of smell.
So the impact can be
widespread.
Widespread.
Not only to the brain itself, but to those nerves that connect the brain to the rest of the body.
And about half of the people that survive end up with
some level of neurological impairment. Yeah. And this can range from mild cognitive impairment to severe physical disabilities.
So the impact can be life-altering.
It can be, yes. And that's, one of the scariest things.
Okay, so let's talk about laboratory findings and the challenges doctors might face when diagnosing. So if somebody comes in with those initial kind of flu-like symptoms, what are the first steps in figuring out if it could be E?
So the initial lab findings in E are often non-specific. So you might see things like an elevated white blood cell count, low sodium levels, and elevated inflammatory markers such as erythrocyte sedimentation rate, or ESR, and C-reactive protein, or CRP.
So those are all signs of inflammation.
Right. They're signs of inflammation.
And not specific to E.
But not specific to EE, they could indicate other conditions.
So those findings alone
wouldn't be enough to diagnose ED.
Okay.
But you might see other changes in the blood as the disease progresses, like a decrease in the white blood cells. Or platelets. And of course, the clinical presentation, the neurological symptoms will also provide clues.
Okay, so those blood tests are like...
Yeah, they're a starting point.
A starting point.
Yeah.
What about analyzing the cerebrospinal fluid?
Ah, yes, the cerebrospinal fluid, the CSF.
Yeah.
So that's often done via a lumbar puncture, which we call a spinal tap. And that analyzes the fluid that surrounds the brain and spinal cord. And in EE, that CSF analysis typically shows elevated white blood cell count, elevated protein levels, normal glucose levels, and in many cases, a predominance of neutrophils.
Okay, so those CSF findings are a little more...
More specific.
Specific.
Yeah.
But are they enough to actually confirm a diagnosis?
So, unfortunately, no. Early on in the infection, it can be very hard to isolate the virus. And even PCR testing might come back negative.
Wait, PCR?
Yeah.
Might come back negative.
It can, especially in the early stages.
Wow.
It's not that the test is faulty. It's just that the amount of virus in the blood or CSF might be too low to detect at that point.
Wow. Okay, so what is the gold standard for diagnosing E?
So once the immune system has had some time to mount a response, serologic antibody testing becomes really key. So that's looking for antibodies that the body has produced against EEV.
So it's like seeing if the immune system has encountered it.
Exactly, even if we can't detect the virus itself.
Wow, okay, so...
And then PCR testing on CSF or brain tissue can also be used to confirm the diagnosis, particularly if those antibody tests are inconclusive.
So again, diagnosing is not straightforward.
It's not straightforward. It can be challenging. And particularly, as we mentioned earlier, in those immunocompromised patients, it can be even harder.
Yeah, because their response might...
It might be muted, So it requires a high level of suspicion from the doctor.
Okay, so what about imaging?
So MRI, magnetic resonance imaging, plays a key role in diagnosing.
And what does an MRI typically show in a patient with E?
So EEV actually has a predilection for deep gray matter structures. Specifically the basal ganglia, the thalami, the mesial temporal lobes, and the brainstem.
And those are deep within the brain.
They are deep within the brain.
And so those areas often show up as those like...
hyperintensities.
Yeah, those bright spots.
Yeah. Yes. Bright spots or hyperintensities. And one of the characteristic findings is symmetric T2 hyperintensities in the lentiform nuclei, which are part of the basal ganglia.
So symmetrical meaning they look the same.
The same on both sides.
On both sides.
Exactly.
And meningeal enhancement, I know, can sometimes be seen.
It can be seen.
But it's
not as common.
Not as common.
In EEE as in some other types of encephalitis. Yeah.
So the MRI not only helps confirm the diagnosis, but also gives us this,
a visual map.
A visual map.
Of what's going on.
Yeah, of what's happening.
Exactly.
And in some cases, that combination of, epidemiological information, patient's presentation, and the MRI findings could be suggestive enough
it could be
to make preliminary diagnosis.
Preliminary, exactly.
Okay, so we talked about MRI. We also briefly mentioned EEG before.
Yes. Electroencephalography.
Yeah, so remind me, how is that?
So the EEG is essentially looking at the electrical activity
of the brain.
And it often shows abnormal patterns.
Yeah, so in EE, it often shows abnormal patterns, including what we call periodic epileptiform discharges.
Okay.
And that's essentially bursts of abnormal electrical activity that can indicate seizure activity.
And seizures, I know, are a common complication.
Very common complication, yes.
RSVE.
And the EEG can help us identify these seizures even when they're not clinically obvious.
Oh, so like what? Silent seizures?
Exactly, silent seizures.
Okay, so the EEG can help us see.
It goes up.
Those?
Yeah.
Okay.
So it's vital not just for diagnosis, but for monitoring and treatment.
Okay.
Yeah.
So we've talked about MRI, EE.
Yeah.
What about the actual pathology?
So EEV targets neurons.
Okay.
And that actually leads to widespread meningoencephalitis.
Okay.
So that's inflammation of both the brain and the meninges.
And the meninges being?
The meninges are the membranes that surround the brain and spinal cord.
Okay.
And so under the microscope, we actually see widespread neuronal necrosis.
So that's death of neurons as well as infiltration of those meninges by inflammatory cells.
So we see this widespread inflammation.
Yes, a lot of inflammation and cell death.
And then also perivascular cuffing.
Yes, so perivascular cuffing.
Remind me what that is.
So that's basically clusters of inflammatory cells around blood vessels in the brain.
And sometimes, you said, we even see
vasculitis.
Vasculitis.
Which is inflammation of those blood vessels themselves.
So it really is a devastating disease.
It is. It is.
And the pathological findings really
underscore that severity of the damage.
Okay, so how do we treat this devastating disease?
So unfortunately, there is no specific antiviral treatment that's proven effective against EE.
So the focus of treatment is really supportive care.
Exactly.
Okay, and what does that supportive care typically involve?
So one of the top priorities is managing those seizures, which we know are a very common complication.
So preventing those seizures to minimize
that further brain damage.
Yeah.
So that involves close monitoring and use of anti-epileptic medications to control that seizure activity.
And then we also need to manage
cerebral edema.
Yeah, cerebral edema. And then
elevated intracranial pressure.
Elevated intracranial pressure.
Yep.
Could you kind of break that down a little bit more?
Of course.
Explain what those things are.
So cerebral edema is essentially swelling of the brain. And that's a serious complication that can occur with any condition that causes inflammation in the brain. Encephalitis included.
And as the brain swells? It puts pressure on the skull, right?
It does. And because the skull is this rigid structure, as the pressure inside increases, it can actually compress the brain tissue.
So how do we manage this swelling and pressure?
So we use a combination of strategies. One is osmolar therapy. And that essentially uses medications to draw fluid
out of the brain tissue.
You can also adjust.
Yeah, we can adjust the respiratory rate. And in some cases, corticosteroids might be used.
So again, a multi-pronged approach.
It is, and all with the aim to protect the brain from further damage.
So that's standard care.
Yes.
Are there any other treatment options that are being explored?
So one treatment that has been used off-label in EE is intravenous immunoglobulin, or IV-RETch.
What is IV-VUG?
So essentially, it is a concentrated solution of antibodies that are collected from the blood of healthy donors.
So we're basically giving them...
Yeah, we're giving the patient...
Pre-made antibodies.
Pre-made antibodies, yes. To help fight off the virus.
And the way IV works is still being researched, right?
It is still being researched, but it's thought to involve a few things. One of them is direct neutralization of the virus by those donor antibodies. Suppression of the inflammatory response. And modulation of the immune system's activity. So it's like a multifaceted weapon.
It is.
And there is some evidence, right? That IVG might improve outcomes.
It might, and particularly if it's given early in the course of the disease.
So it really is a two-pronged attack.
It is.
Making E prevention that much more challenging.
It really highlights how important it is to understand their ecology and behavior. So we can develop effective control strategies.
It's not just about killing mosquitoes indiscriminately.
No, we need to target the species that are most likely to spread the virus to us.
And so I know we touched on climate change before.
Yes.
But, we're seeing biodiversity loss and habitat fragmentation. How are those things?
Yeah, so biodiversity loss, and that essentially means a decline in the variety of species in an ecosystem.
That can actually increase the risk of disease transmission.
It seems counterintuitive.
Yeah.
Right. You would think fewer species, fewer opportunities, but that's not always the case.
Okay.
So in a diverse ecosystem, you have a complex web of interactions between those species.
Okay.
And that complexity can actually help regulate disease transmission.
So like, for instance, some predators might specialize in eating certain mosquito species.
Exactly. That helps keep the population under control.
So it's like a natural...
It is. It's nature's checks and balances.
Yeah, okay.
But when we lose that biodiversity, we disrupt that balance.
And that can lead to a proliferation of species that are more likely to transmit.
Right, like those bridge vector mosquitoes.
So basically, by simplifying these ecosystems,
a
lot of times through our activities...
through deforestation.
Urbanization,
we're creating conditions that are more favorable.
And then we have habitat fragmentation.
Yes, habitat fragmentation.
What is that, and how does that play a role?
So essentially, habitat fragmentation is the breaking up of these large, continuous habitats into smaller, isolated patches.
So you're taking like...
Yeah, think of a forest.
A big forest, and you're like...
Dividing it up.
Yeah, dividing it up.
With roads, buildings.
Okay.
And so when this happens, the animals are often forced into closer contact with each other.
And this...
Yeah, so it makes it easier for diseases to spread.
So biodiversity loss and habitat fragmentation
are really important ecological factors that can increase that risk of transmission.
Wow.
Yeah, it's a reminder that our actions have these ripple effects.
So we really are a part of nature.
Yeah, we are.
Okay, so we talked about the devastating effects on individuals and families.
Yeah.
But how does this translate into, like...
Yeah, so any serious infectious disease is going to have these ripple effects that extend beyond those individual cases. It impacts communities, healthcare systems, and even entire economy.
Wow, can you give us some examples?
Of course.
Yeah.
So outbreaks can put a significant strain on healthcare resources.
Okay.
Hospitals can be overwhelmed with patients.
So, it's not just about treating...
Right, we have to make sure that the entire healthcare system is able to function effectively.
Yeah, for everyone.
Yeah, for everyone.
Okay.
And then, of course, beyond healthcare, there are the economic effects.
Okay, how so?
So there's the lost productivity as people miss work because they're sick or they're caring for sick family members.
Okay.
This can impact businesses, particularly small businesses.
Yeah.
And then there are the broader economic effects.
Okay.
Because people might avoid going out.
Yeah, because they're afraid.
Exactly.
Yeah.
And this can impact businesses that rely on tourism, recreation, entertainment.
And then there's the cost.
The cost of mosquito control.
Yeah.
Public health campaigns.
Yeah, all of those efforts.
All of those things really add up.
Yeah.
And become quite costly.
And then the cost of fear and anxiety.
Right. So there's the intangible cost.
So it really is a reminder that these infectious diseases aren't just medical problems. They're
social problems. They're economic problems.
Yeah, it's all connected.
Exactly.
Okay, so let's discuss the ethical considerations.
Right. So anytime we're dealing with a disease like this, we have to think about the ethics.
What are some of the ethical dilemmas?
So one of the most fundamental ethical considerations is this balance between individual rights and the public good.
And so how does that play out with E?
So imagine there's an outbreak.
Okay.
Public health officials may recommend or even mandate things like mosquito spraying, quarantines, travel restrictions.
So it's about
It's about finding that balance.
Yeah, finding that balance.
Between protecting the public and not infringing on individual freedoms.
And then we have
the allocation of resources, which are often very scarce during an outbreak.
So we have to figure out.
We do. We have to have guidelines in place to distribute fairly and equitably.
And, then it gets even more complicated when you have...
Right. Social and economic disparities.
Yeah.
Absolutely. So what about the research?
Right. So all research involving EE needs to be conducted ethically and responsibly.
And so when we're talking about animal models,
yes,
which are, really essential.
They are essential, but we have to make sure that we treat them humanely.
Yeah, and minimize
suffering.
Yeah.
Yeah.
And then research involving human subjects.
So informed consent is absolutely paramount.
And confidentiality.
And confidentiality is key.
Yeah.
So we have to protect their information.
So again, it's
really about
conducting this research responsibly with integrity.
Exactly. Putting those participants first. So we've covered a lot of ground today.
We have.
From the virus, how it spreads, clinical presentation, treatment research.
Yeah.
What are your final thoughts?
So I think the key takeaway is that EE might seem like a rare, obscure disease.
Yeah.
But it is a very serious public health threat.
Yeah.
And we really need to be prepared for it.
Yeah.
While we've made great strides in understanding it, there's still a lot.
We still have...
Yeah, we still have a lot to learn.
Yeah.
And as our climate changes, human activities continue to impact the environment, the risk of outbreaks is likely to increase.
That is a very sobering...
It is, but it's not all doom and gloom. We do have the tools and the knowledge.
Yeah.
We need to be proactive about mosquito control, educate ourselves, support the research.
So it's really about,
It's about awareness, prevention, research, and it's a collective effort, all of us, really working together to protect ourselves and our future.
And it's really about the people.
It is.
Right.
Absolutely.
We have to remember the humans.
Yes, it's a human problem. We have to approach it with compassion.
Yeah, with empathy.
Absolutely.
Well, from Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.